Hey guys, Mrs. Yunker, and today we are talking about the nature of science and the scientific method. So when we start talking about experiments versus other investigations, experiments have a hypothesis, an independent variable, a dependent variable, controlled variables, and they can establish cause and effect relationships. They also use the classic scientific method. So when we start talking about science fair, that's the kind of thing we're doing. It's a full-blown experiment. All of those pieces are there. Other investigations like lab demos, models, some hands-on activities we do, those are just scientific investigations. They might be guided by a question. They don't necessarily follow the whole scientific method. They may not have a hypothesis, and you can't really establish cause and effect relationships from them. So the whole scientific method, this is what we're talking about. When we're following the scientific method, we start by asking that question, doing some background research, conducting a hypothesis, and then we get to move on to test our experiment. If our procedures are working, we can keep going. If they're not working, we might have to go back and troubleshoot the procedures, make sure that we followed them all. If there's something weird in our procedures, we might have to tidy up our procedures and restart. I've seen students who are growing like grass seed. They decided they were going to put 100 milliliters of water on them every day. And by day 3, they are drowning in mold, and they are going to learn nothing from that experiment. So it's a time where we can kind of throw those procedures out, write new procedures that are more appropriate, and fix that problem, and then start experimenting. So we don't just waste 2 weeks of our time. So once we have our procedures working, we're going to analyze data and draw conclusions. When we do that, if our results support our hypothesis, we communicate those results. If our results only partially support or fail to support our hypothesis, we still communicate those results. However, we can also go back and use that experimental data that we now have. It becomes our background research for new or future projects, and we can ask a new question, form a new hypothesis, and experiment again. So scientific method, things are always kind of mixed up and going back and forth. All right, remember, whenever we see the pencil, we need to write things down. This whole slide has lots of valuable information we're going to be referring to all year long, so please copy down the entire slide. You can pause it at any point that you need to. So we're talking about our test variable, which is our independent variable. This is the variable that is changed by the experimenter. There can only be one independent variable. So there's only one of them, and I change it. Independent variable, I change. I know it sounds ridiculous, but it really helps you remember it. So I would recommend going, I change. Maybe when you're alone and no one's watching. All right, outcome variable or the dependent variable depends on the test variable and is the variable being measured. So if the independent variable is what I'm watering plants with, the dependent variable is how far they grow or how much they grow. Your controlled variables or your constants, these are the things that are kept constant. Constant, capital S, same. Things like temperature, amounts, methods, you're doing the same thing to all of your plants. You're watering with the same amount of liquid, even if it's a different liquid. Your control group, be really careful with these two. People tend to get them mixed up. The control group is a standard used to compare to, a group that doesn't receive what is being tested. Not all experiments have a control group. Strong experiments usually do, though. So if you want to give some good strength to your results, you want to make sure you have a control group. So in the experiment, if we're talking about watering plants with different liquids, the control group would be water, because that's what we normally give plants. Again, feel free to pause if you need to still copy those down. Alrighty. Hypothesis. A hypothesis is the proposed explanation. This is how we normally write it up. I need you to copy down the definition and this part, okay? If, with the independent variable, then what do we think is going to happen to that dependent variable? Because, and we give some scientific reasoning, some evidence, some research there. So here's an example. If we test the pH level of lemon juice, baking soda, and orange juice, those would be my independent variables, then the orange juice will have the highest pH. That would be my dependent variable, because more people report having heartburn after drinking orange juice. I don't know if that's true. I made that part up. Just for an example. When you are done with your experiment and you're looking at results and data, you can say that your results support or results fail to support your hypothesis. Please write that part down also. Neither one is wrong. We can learn from both of them and lead to further testing when you have a strong experiment. Data. We've already talked a little bit about empirical data. Remember, scientists collect empirical data, the data we cannot argue with, the empire, things we can see and measure and all agree that that's what happened. While you're doing an experiment, you should be following your procedures and taking data in a table, which means in your procedures, there should be a place that tells us how often you're collecting that data and what data you're using. You're going to use metric units like meters, liters, Celsius, and grams. And this is what a chart will look like. Please copy down the example of a chart so that you have this to refer to when we get to science fair and those kinds of things. At the top, there should be a title, which is the purpose of your experiment. Down this side, we should have our independent variables and we should write out the units. Across the top, we have dependent variables and units, and then we have trial 1, trial 2, trial 3. Remember, repetition is multiple trials. So that's why we do multiple trials, to make sure that our data is accurate, and maybe an average or things like that on the side. And so here's an example chart while you copy that one down. The title is the pH of common household substances. I have my substances listed down the side: lemon juice, baking soda, orange juice. These are my pHs, trial 1, trial 2, trial 3, and the average, and all the numbers are filled in. So that is what a chart looks like. If you were going to take this information to a graph, which we often need to do, then on the independent variable, so for this example, it would be the substances, goes on the x-axis on the bottom, independent on the bottom, and the dependent variable goes on the top because it's what we're measuring, so it's what's going to change. Finally, after you've done your chart, after you've done a graph, then we start talking about our results. The results section, when we're talking about an experiment, is not where you write your conclusions. It's not. It is where you take all of that lovely data and you write it in words. You might give some little anecdotes about things that happened, but you're mostly just giving us this information in words. That's it. This is not where you're writing your conclusions. So let's go ahead and talk about our conclusion. The very last, before we can get to communicate those results, is to write our conclusion. You're going to summarize your experiment in a few sentences. You're going to state whether your results support or fail to support your hypothesis. And you're going to restate that whole hypothesis there. Based on this data, my data supports my hypothesis because, and you're going to kind of give all that information again. You're going to summarize and evaluate your procedures. What worked, what didn't work. Did you have to redesign your procedures and start over? That's the information you're going to add there. And suggest changes to experiment and possible for study. That part's a lot easier if your results don't fit your hypothesis. But we can always go ahead and look about, well, what could I learn more about? After the findings are communicated to other scientists could replicate your results. Again, I need you to write these two down. Repetition is repeated trials by the same experimenter, trial 1, trial 2, trial 3. And replication is the experiment is copied by another scientist or experimenter. It helps to validate the experiment. So we want to be able to repeat our trials. And the more repeated trials you have, the better your data is. And you definitely want someone else to be able to replicate and get similar results than you did. All right, so that's all I have for you today. Have a great day.